Okay, so welcome back to this next video in which we are discussing the GABA-A receptors. Okay, right, so in the previous video what we saw is that there are these three superfamilies of ligand-gated ion channels, okay, and we've seen that the GABA-A receptors all fall into this final superfamily of ligand-gated ion channels, which are the nicotine-like receptors, which would have once been called the cis-loop ligand-gated ion channels, and in fact, everyone still calls them this. Okay, although their official name is now the nicotine-like receptors. Okay, right, so that means that all GABA-A receptors are pentamers, okay? They're made up of 5 separate subunits joined together to make the full ion channel, okay? And each of those subunits that makes up a fifth of a GABA-A receptor has the same basic structure, okay? So it starts with its amino terminus extracellularly. It then has this very distinctive structure within its extracellular portion known as a cis loop, which is a loop in the polypeptide structure held together by a disulfide bridge between two cysteine residues that are 13 amino acids apart. Okay, then you have the first membrane spanning alpha helix, a second membrane spanning alpha helix, a third membrane spanning alpha helix, then a large intracellular loop between the third and then the fourth membrane spanning alpha helix, which then has the carboxylic acid terminus extracellularly. Okay, so you take 5 proteins which all look like this, stick them all together, one here, one here, one here, one here, and one here, and then you end up with the full GABA-A receptor. Now, as I said in the previous video, what would be beautifully simple is if there was one gene which coded for the GABA-A receptor subunit. You use this gene to make 5 copies of this protein which were all identical. You stuck the 5 proteins together and you have then got a GABA-A receptor. It is not so, unfortunately. There are instead 19 different genes for GABA-A receptor subunits. Okay, right. So these all code then for 19 different proteins which will have different sequences of amino acids. Now, these 19 different proteins will all have the same basic structure. They'll all have the cis loop, they'll all have the same membrane spanning topology, okay? But they will all have different sequences of amino acids. So let me now go through the names of the 19 different subunits that you can use to make up a fifth of a GABA-A receptor. Okay, so for the first part, they're divided into 4 families. Okay, so the first family is the GABA-A receptor alpha subunits. Okay, and I'll often use this notation now for GABA-A. Okay, so previously I've been showing GABA-A with a dash here. Okay, that's because I can write that in the YouTube video title. But often people, when writing GABA-A receptors, they'll put the A as a suffix down here. Okay, so these are the GABA-A receptor subunits alpha. Okay, now this is a whole family of GABA-A receptor subunits, and there are 6 of them. Okay, so there is the GABA-A receptor subunit alpha type 1, then there is the GABA-A receptor subunit alpha type 2, okay, and it goes on. So then there's alpha 2, alpha 3, alpha 4, and it's overall you have 6 of the things. So then there's alpha 5, and then finally alpha 6. So, there are first 6 different GABA-A receptor subunits. So we've done 6 now, so we've got 13 more to go of the 19. Okay, so that's the first family of GABA-A receptor subunits. Okay, the next family then. Okay, the next family is called the GABA-A receptor subunits beta. Okay, the beta family of GABA-A receptor subunits. And this contains 3 different GABA-A receptor subunits. It contains the GABA-A receptor beta 1 subunit, okay, the GABA-A receptor beta 2 subunit, and then finally also the GABA-A receptor beta 3 subunit. Okay, so that now takes us down to 10. We've done 9 different GABA-A receptor subunits. Okay, so these are 3 more grouped into a family together. Okay, and you'll see the point of grouping them into families like this in a moment. The next main family, and these first three families we're looking at, these are the main three: the GABA-A receptor subunit alphas, the GABA-A receptor subunit betas, and the GABA-A receptor subunit gammas. Okay, the other ones are a little bit more niche and specialized. Okay, right, so the GABA-A receptor subunit gammas then. Okay, again there are three members of this family: the GABA-A receptor subunit gamma 1, the GABA-A receptor subunit gamma 2, and the GABA-A receptor subunit gamma 3. Okay, so that's now 3 more, so overall we've done 12 now, and we've got 7 more subunits to go. Okay, so there is one more family of GABA-A receptor subunits before they then go in on their own families, basically. Okay, so the next family that has multiple members in with it is the GABA-A receptor family Rho subunits. Okay, and there are 3 of these. These are called the GABA-A receptor subunit Rho 1, the GABA-A receptor subunit Rho 2, and then the GABA-A receptor subunit Rho 3. Okay, right. So these are the 3 subunits of GABA-A receptors that are in this Rho family. Okay, now the rest of them, okay, and how many have we still got left to do? We've got 4 left to go. The final 4 are in families by themselves, basically. So, there is the GABA-A receptor subunit epsilon, okay? Then there is the GABA-A receptor subunit delta, okay? Then there is also the GABA-A receptor subunit theta, and then finally the GABA-A receptor subunit pi. Okay, so we're just exploring the Greek alphabet a little bit more. So those are the final 4 subunits of GABA-A receptors. Okay, right. Now, as I say, if you're going to remember a few of them, remember the alphas, the beta, and the gammas. Okay, these ones are more niche. You find them in specialized places, usually. Okay, right. So those are 19 different subunits, then, of GABA-A receptors. Okay, now, that then means that there is this horrendous complexity with regards to building GABA-A receptors. You have 19 different subunits. To build a full GABA-A receptor, you need to stick 5 of these subunits together. The question then arises, how do you stick them together? Okay, well, there are multiple, well, clearly there are thousands of different GABA-A receptors that you can build, but generally GABA-A receptors conform to a certain structure, basically. They use certain types of subunits. So let me now tell you about these different conformations, basically. Okay, so most GABA-A receptors are what are known as alpha, beta, gamma GABA-A receptors. Okay, and this is why I said that the alpha family, the beta family, and the gamma family were the main families of GABA-A receptor subunits that you should know. Okay, because these GABA-A receptors are going to contain alpha subunits, beta subunits, and then gamma subunits. Okay, right. So let's now explore these alpha, beta, gamma GABA-A receptors. So we are now looking at the GABA-A receptor from above. So imagine looking down like so at the top of the GABA-A receptor from the outside. Okay, and we want to look at the arrangement of the subunits. Okay, so it's a pentamer. It's made up of 5 separate subunits joined together like so. Now, in these alpha, beta, gamma GABA-A receptors, you're going to have 2 alpha subunits, 2 beta subunits, and 1 gamma subunit. So let me show how they're going to be arranged. So you're going to have a beta subunit here, an alpha subunit here, another beta subunit here, an alpha subunit here, and a gamma subunit here. And once again, I will stress we are looking from the outside of the cell down at the GABA-A receptor. So going clockwise round, you have alpha, beta, and then gamma. Okay, right, so let's colour in these different portions. So we'll colour in the 2 alpha subunits of our alpha, beta, gamma GABA-A receptor in green here. Okay, we'll colour in our 2 beta subunits of our GABA-A receptor in orange here, okay, and then we'll colour in our gamma subunit of our GABA-A receptor in blue here. Okay, now that's not the complete story, because there are multiple alpha subunits, there are multiple beta subunits, and there are multiple gamma subunits. So which ones am I going to put in here? Well, I'll show you the main type of GABA-A receptor that you have in the brain in a moment. But in general, the rules for building a GABA-A receptor, and I've just realised I haven't colour-coded this at all. The alphas here are in a complete different colour. I just coloured those in randomly. I'm sorry about that. Okay. Basically, the rules for building one of these alpha, beta, gamma GABA-A receptors is you firstly go to this GABA-A alpha family of GABA-A receptor subunits. You pick one of the 6 members, so choose one of the 6 members, okay? You will use that member in these 2 slots here. You'll use it here, and then you'll use it here, okay? So you don't use 2 different alpha subunits here. It's always the same alpha subunit, okay? So you pick the same alpha subunit, and you use it twice in your GABA-A receptor here. You then go to the beta family of GABA-A receptor subunits. You pick one member from this family, and you use it again twice in these 2 slots here. You put one there, one there. Then you go to the gamma family, pick one, and you put it there. Okay, and that's how you build one of these alpha, beta, gamma, GABA-A receptors. Okay, so now to tell you about the most important GABA-A receptor that you find within the brain. So the main one contains as the alpha subunit, alpha 1. So both of these subunits will be GABA-A alpha 1 subunits of GABA-A receptors. Okay. The beta subunit is then beta 2, so both of these will be GABA-A beta 2 subunits of GABA-A receptors, and finally the gamma subunit will be a gamma 2 subunit of a GABA-A receptor. Okay, right, so this is the main form of the GABA-A receptor that you find within the brain. Okay, so if you're going to learn one GABA-A receptor, this is the one to learn. Okay, right. Now, let's talk about where the GABA binding sites are actually located on these alpha, beta, gamma GABA-A receptors, and specifically on this main alpha 1, beta 2, gamma 2 GABA-A receptor. Okay, so basically each GABA-A receptor of this form only has 2 GABA binding sites, okay, so not 5 GABA binding sites, just 2, okay, and these are located in between the alpha and the beta subunits. So you'll have one here, okay, between this alpha subunit and this beta subunit, and then you'll have one here between this alpha and this beta subunit, okay, so these are the 2 GABA binding sites, so GABA will bind here basically. Okay, so to open the GABA-A receptor, what has to happen is a GABA molecule has to bind here, and a GABA molecule has to bind here as well. So 2 GABA molecules are required to open the GABA-A receptor. Okay, right, and since I've made it plural, I need to have an arrow to the other one as well. Right, so that then is the GABA-A receptor structure. What we now want to talk about is what's actually going to happen once the GABA-A receptor opens. Okay, so you will have these receptors on neurons. Okay, most of them will be located in dendritic spines on the postsynaptic membrane, which we'll talk more about in a moment. Okay, some of them will be located extrasynaptically, and we'll talk about those as well. But for now, what we want to talk about is when GABA binds to the GABA-A receptor and the GABA-A receptor opens, what, firstly, is it going to allow to move through it? Well, I can give you the answer to that now. It's chloride anions, okay? And what effect is that going to have on the electrical potential difference across the cell membrane? Okay, so to discuss this, let's start off with a discussion of electrical potential difference across a cell membrane. Okay, so to discuss electrical potential difference, we firstly want to discuss a topic called electrical potential, which is very much so related but is more foundational than electrical potential difference. Okay, so electrical potential then. Electrical potential is a concept from physics. Okay, it is a mathematical model to help us understand this reality which we live in. Okay, and this mathematical model goes like so. Okay, so this is my attempt to draw a three-dimensional graph. To every point in three-dimensional space, you are going to ascribe a real number. So you're going to go to every single point in three-dimensional space, and you are going to give that point a real number. Okay, now this real number can be positive, it can be 0, it can be negative, it can be an integer, it can be a rational, it can be an irrational, it can be any real number you like. The limitations are that you're not allowed to go outside of the real number system. You're not allowed to go into more complicated number systems like complex numbers. You have to remain in the real number system. Okay, so you're going to ascribe to every point in three-dimensional space a real number. Okay, and that real number that you've ascribed to every point in space is going to be called the electrical potential. Okay, now you can't just make this up. Every point does have an electrical potential, and there is some way of working out what number every point in three-dimensional space should have. But that's the basic idea, that every point in three-dimensional space has this real number attached to it that in principle you can work out. Okay, right. Now, electrical potential is measured in volts. Okay, so that's the units of electrical potential. So, in principle, what I can have is a little man here, okay, with a machine here, which is capable of measuring electrical potential. And this little man can go and stand anywhere in three-dimensional space, and he can ask, what is the number on my machine? i.e., what is the electrical potential of this point? Okay, so that's electrical potential. Let's now take this back to biology. Okay, so let's have a cell here. Okay, so this is a cell. Now, all of the points inside the cell, i.e. all of the points of the cytoplasm, to a rough approximation can be considered to all be in electrical equilibrium, okay, and therefore are all going to have the same electrical potential. So this number that's ascribed to all of the points inside the cell is going to be the same. So this little man could go inside the cell. Okay, so he could go in here and he could wander around and the number on his screen would not change. Okay, it would remain the same. Let us call the number that would stick on his machine, we'll call that the electrical potential, big E, and then we'll subscript it I for intracellularly. Okay, so this is just the number that this man would have on his screen wherever he stood in this cell. Okay, right. Similarly, he can go into the extracellular fluid, okay, and he can ask what is the electrical potential there, okay, and again, if he wandered around anywhere in the extracellular fluid, okay, so in all of this extracellular fluid, he would get the same number on his screen because all of the points of the extracellular fluid are in electrical equilibrium with one another. Okay, we'll call the value that he would have on his screen the electrical potential, capital E, and then we'll subscript it E for extracellularly. Okay, right. Now, the question is the electrical potential of all of these points in the extracellular fluid the same as the electrical potential of all of these points in the intracellular fluid? And the answer, as you can probably guess, is no. Okay, they're not. So then comes the concept of an electrical potential difference, okay, across the cell membrane. Now, the other name for an electrical potential difference is a voltage, okay? So, when people say the voltage across the cell membrane or the electrical potential difference across the cell membrane, strictly speaking, what they should say is the electrical potential difference from extracellular to intracellular. They should give a direction in which you are moving. People often drop this, okay? They assume that you understand that they always mean going from the outside of the cell into the cell, because usually experimental physiologists, they start off outside the cell and they then have to go into the cell. They then have to stick their electrode into the cell in some way. So it's assumed that you always implicitly understand that it is from extracellular to intracellular. But strictly speaking, they should say the voltage from extracellular to intracellular. Now, what does this mean? Well, this means the electrical potential difference that you would feel if you moved from extracellular to intracellular, or stated even more basically, it means if this little man starts off in the extracellular fluid, okay, so he's currently standing here, okay, he will have a number on his screen, and this number will be the electrical potential extracellularly. Now, if he somehow gets himself from the extracellular fluid into the intracellular fluid, there will now be a different number on his screen, which will be the electrical potential intracellularly. The electrical potential difference when you move from extracellular to intracellular means how much does this number on his screen change when he moves from extracellular to intracellular. Okay, so what is the answer to that in terms of the electrical potential extracellularly and the electrical potential intracellularly? Okay, well, it's going to be the number after he's moved. Okay, so the second number, okay, the number that it's going to become, which is the electrical potential intracellularly, subtract off the original number that was on his screen, which is the electrical potential extracellularly. Okay, so this tells you how much the number on your screen changes when you move from extracellular to intracellular. Okay, right, so that's what is meant by the electrical potential difference from extracellular to intracellular across the cell membrane. Okay, right, and the answer to this is generally around negative 65 millivolts. Now, firstly, let's discuss the fact that the electrical potential difference across the cell membrane is going to be measured in the same units as electrical potential, okay, because we are just taking one electrical potential and subtracting off another electrical potential. That is not going to change the units that we are working with here. The difference between two electrical potentials is also going to be measured in volts. Millivolts just means 1,000th of a volt, okay, so really you could write this as negative 0.065 volts. Okay, so what that literally means is that the electrical potential intracellularly is lower than the electrical potential extracellularly, and it's lower than the electrical potential extracellularly by a whole 65 millivolts. Okay, so that's what is meant by the electrical potential difference across the cell membrane. Now, let's now discuss what's going to happen when we open a GABA-A receptor by binding GABA to those two GABA binding sites on the GABA-A receptor. Okay, so here is our pentameric GABA-A receptor sitting in the cell membrane. Here is the ion channel down the middle. Okay, right, so let's suppose that two GABA molecules have come and bound to the GABA-A receptor and have now triggered it to open. Okay, so it's now opened. It's now an open chloride anion channel, so chloride anions can move through it. Okay, the question is, which direction are they going to move? Okay, well, there are two things we need to consider here. We need to consider the concentration gradient of chloride anions across the cell membrane, and we also need to consider the electrical gradient across the cell membrane. Okay, because chloride anions are a negatively charged particle, they will feel the electrical potential difference across the cell membrane. Okay, they will be influenced by that. So, we know what the electrical potential difference across the cell membrane is. It's negative 65 millivolts, which means that the intracellular compartment is 65 millivolts lower than the extracellular compartment. Now, it's a general rule of physics that negatively charged particles want to go where the electrical potential is higher. Okay, so chloride anions, if they were just listening to the electrical gradient, they would want to go out of the cell. So they would want to move out, okay, but they're not just going to listen to the electrical gradient, because the concentration gradient is going to have a say as well. Okay, so now we need to discuss what the concentration gradient of chloride anions across the cell membrane is. Okay, well, usually chloride anions are higher extracellularly than they are intracellularly. Okay, however, for different cells and at different times, it will differ what this concentration gradient is, okay? So sometimes the concentration gradient will be not that strong, whereas other times it will be much stronger. However, usually it always is that chloride anions are higher outside the cell than inside the cell, and that means that the concentration gradient is always going to be saying, go into the cell. Now, we have the electrical gradient saying one thing and the concentration gradient saying another thing. So the question now is, who wins? Who is stronger? Now that will depend on how large this concentration gradient of chloride anions across the cell membrane is. Now, if the chloride anion concentration gradient is greater than 11 times. Okay, so what I mean by that is if this chloride concentration extracellularly is greater than 11 times bigger than the chloride concentration intracellularly, i.e. this number is more than 11 times bigger than this number, then that concentration gradient will be strong enough to mean that the driving force for chloride anions to go into the cell will beat the driving force for them to leave the cell because of the electrical gradient, and you will overall get a net movement of chloride anions into the cell. Okay, so that's the usual situation, that the chloride concentration gradient across the cell membrane is strong enough to completely beat the gradient with regards to the electrical potential, which is telling the chloride anions to go out of the cell. Okay, and you'll therefore get a net movement of chloride anions into the cell once you open this GABA-A receptor. However, in some cells at certain time points, okay, the concentration gradient can be less than 11. Okay, so this one, let's say it could be 9 times bigger than this one. If that is the case, then the force driving the chloride anions into the cell will be too small to beat this force that's trying to push them out, and in that case you will actually get a net movement of chloride anions out of the cell. And these two effects, one where the chloride anions are coming into the cell and one where they're going out of the cell, these are going to have completely, and I'll repeat this, completely opposite effects on the electrical potential difference across the cell membrane. Now, let me stress that for most cells in the brain, the chloride concentration gradient across the cell membrane is going to be greater than 11-fold, okay? Which means that overall, when you open the GABA-A receptors, you are going to get a net movement of chloride anions into the cell. But sometimes, in some cells, the concentration gradient will be less than 11, and in that case, you're actually going to get a net movement of chloride out of the cell. So, we're going to discuss these two opposite effects and the opposite effects that they're going to have on the electrical potential difference across the cell membrane, and then we'll put it into context with regards to what it means regarding action potential generation, because we'll see that a chloride current coming into the cell is going to be inhibitory. It's going to promote you not firing an action potential, whereas a chloride current going out of the cell is actually going to be stimulatory, so it's going to have complete opposite effects. Okay, but more normally, you're going to get a chloride current coming into the cell. Now, the reason I'm referring to it as a current is a current just means a movement of charged particles. These chloride anions are negatively charged, so it is a movement of negative charge into the cell, so it is a current. Okay, right. So if you've got a current of chloride anions coming into the cell, what effect is that going to have on the electrical potential difference across the cell membrane? Well, When you move negative charge into an area, that is going to reduce the electrical potential of that area. Okay, so we are bringing chloride anions into the intracellular fluid, therefore we are going to reduce the electrical potential intracellularly. Okay, so this number is going to go down. In addition, we are taking chloride anions out of the extracellular fluid. Okay, so when they were in the extracellular fluid, they will have been making the electrical potential lower. Okay, so once you remove them out of the extracellular fluid, that will basically, it's like a spring being compressed and it will come out, it will, recoil. So once you've removed the chloride anions from the extracellular fluid, that will make the extracellular fluid's electrical potential go up. So the electrical potential extracellularly is going to go up, the electrical potential intracellularly is going to go down, and that's because of this inward current of chloride anions shown here in purple. Okay, now what effect is that going to have on the electrical potential difference across the cell membrane? Well, the electrical potential difference across the cell membrane was originally negative 65 millivolts, which meant that the electrical potential intracellularly was 65 millivolts lower than the electrical potential extracellularly. Okay, we have now raised the electrical potential extracellularly, and we've lowered the electrical potential intracellularly. So these two numbers are going to be moving further apart. So if I show this on a graph, well, just in a diagram here, let's say that this represents the electrical potential extracellularly originally, and this is the electrical potential intracellularly. And originally you had to step down by negative 65 millivolts when you moved from extracellular to intracellular. Now we're making this number go up, okay? We're making this number go down. So the amount that you're going to have to step down when you move from extracellular to intracellular is going to get bigger, okay? So this number is going to become more negative, basically. Okay, I'm rapidly running out of space here. And the name for a current, well, the name for the electrical potential difference across the cell membrane becoming more negative is hyperpolarization. So we're going to hyperpolarize the cell membrane by opening these GABA-A receptors, basically. Okay, right. Now, it's going to be completely the opposite effect. Oh, actually, before I talk about the complete opposite effect, let me give you a little bit more nomenclature. It's just I haven't got enough space to actually write it down, so I'll get another piece of paper here. So, this hyperpolarization of the electrical potential difference across the cell membrane has another name. It's also known as an IPSP. Okay, and what this stands for is an inhibitory, because it's going to inhibit the postsynaptic neuron from firing an action potential. Postsynaptic, because it's acting on the postsynaptic cell, okay? And then finally the P is for potential. So an IPSP stands for an inhibitory postsynaptic potential, which is another name for this hyperpolarization of the electrical potential difference across the cell membrane that is going to occur because of the opening of these GABA-A receptors. Now, one final piece of terminology I'd just like to talk about, which is very much so related to this, is the concept of an IPSC. Okay, so the IPS stands for the exact same thing. It stands for inhibitory postsynaptic. Okay, but now the C stands for current. Okay, so what is an inhibitory postsynaptic current? Well, this refers to that chloride anion current that we have coming into the cell. Okay, so remember I explained why I was calling this a current, because it's a movement of charged particles, but because it's going to inhibit the postsynaptic cell, it's also known as an inhibitory postsynaptic current, or an IPSC. So the current is called an inhibitory postsynaptic current, or an IPSC. The change in electrical potential difference that this produces, the hyperpolarization, is then called an IPSP, for inhibitory postsynaptic potential. Okay, now, the other situation which occurred when the concentration gradient of chloride anions across the cell membrane was not big enough to counteract the force of the electrical gradient, which is trying to force chloride anions out, and you therefore get a net movement of chloride anions out of the cell. Okay, this is going to have the complete opposite effect on the electrical potential difference across the cell membrane. Okay, we are going to be raising the electrical potential intracellularly. Okay, because we are moving negative charge out of the intracellular compartment, and therefore the electrical potential intracellularly is going to go up. Okay, this time, however, we're going to be lowering the electrical potential extracellularly because we're moving negative charge into the extracellular compartment. So on this graph, we're going to be making this number here go down. Okay, and we're going to be making this number here go up, so we're going to be making the drop down smaller, basically. We're going to be making the electrical potential difference more positive. Okay, and the fancy word for making the electrical potential difference across the cell membrane more positive is depolarization of the electrical potential difference across the cell membrane, and it has an analogous name to this. It's also known as an EPSP. Okay, and this time this stands for an excitatory postsynaptic potential. Okay, right, and there is similar terminology for the actual movement of chloride anions out of the cell. It's now known as an EPSC for an excitatory postsynaptic current. So this movement of chloride anions out of the cell, this would be called an EPSC for an excitatory postsynaptic current. Okay, right. So that's the effect that opening these GABA-A receptors is going to have on the electrical potential difference across the cell membrane. And once again, I will stress that in nearly all cases, it's going to be that the chloride anion concentration gradient is big enough to overpower the electrical gradient, and therefore you're going to get this IPSC coming into the cell when you open these GABA-A receptors, which will therefore hyperpolarize the electrical potential difference across the cell membrane and cause and make it less likely that the neuron is going to fire an action potential, making GABA predominantly an inhibitory neurotransmitter, and it's usually referred to as the most important inhibitory neurotransmitter within the brain. Okay, however, occasionally it can be excitatory because the chloride anion concentration gradient is not big enough to overpower the electrical gradient driving chloride anions out. But that's very, very rare that that occurs. Okay, but if you do see that, that's what's happening basically. Okay, right, so the final thing that I now want to discuss is to put it all into context, because I've described how the GABA-A receptor allowing chloride anions into the cell, so we'll assume the more normal thing is happening where you've got a high enough concentration gradient of chloride anions to overpower the electrical gradient which says move in the opposite direction. Okay, so what we now want to see is how this inhibitory postsynaptic current is going to inhibit the firing of an action potential within the postsynaptic neuron. I'd also just like to talk about extrasynaptic GABA-A receptors as well. Okay, right. So for this, let's draw a neuron here. Okay, so the main landmark features then of a neuron are we have the dendrites here coming off the cell body. So here is the nucleus right at the center here. Then we've got these processes coming off the nucleus known as the dendrites. Now I'm going to draw a little process that now comes off a dendrite, which is known as a dendritic spine. Okay, so each of the dendrites will have loads of these little dendritic spines coming off them. And I'll discuss them more in a moment. Okay, then we have the axon here. And then right at the end of the axon, we'll have the axon terminal, which I'll show here. Okay, right, and we'll put a few myelin sheaths here. So here is a myelin sheath, here's a node of Ranvier, here is another myelin sheath. Okay, right, so this is the cell body of the neuron here, called the soma. Okay, these are the dendrites here. okay, which are these processes coming off the cell body, and you have multiple dendrites. This other long process that comes off the cell body, and unfortunately on this picture I haven't really drawn it that much longer than the dendrites, but it will in reality be much longer than the dendrites. This is known as the axon, and each cell only has a single axon, or at least usually only has a single axon. Okay, and then each of the dendrites is going to have these dendritic spines coming off. Now, I've only actually shown a single dendritic spine here, okay? But in fact, all of these dendrites are going to be covered in these tiny little processes coming off them called dendritic spines. And it is these dendritic spines that receive the axon terminals of other neurons, okay? So this, drawn rather large here, it's drawn out of scale here. Okay, this is the axon terminal of this large neuron. Okay, here is the axon terminal of another neuron here. Okay, now this axon terminal from the second neuron is going to be having an input onto the dendritic spine of this next neuron along. So let's colour in the portions here. So we have our dendritic spine in green there. Okay, we'll colour in axon terminals in blue. So here's an axon terminal in blue. Okay, right. I'll colour in the myelin sheaths in yellow. Okay, so these are just speeding up the transmission of the signal along the axon. Okay, because they allow, remember, the action potential to jump from node of ROMVA to node of ROMVA. Okay, so these are myelin sheaths. Okay, what else can we colour in? Oh, another very important thing to mention is that there is this region that is just at the start of the axon, and this is going to be extremely important, and people often forget about this, but this is an extremely important concept. It's the Axon Hillock, and this is where the action potential is actually going to be generated. Okay, so now let me tell you about how you cause this neuron here to fire an action potential. Okay, so this neuron is going to have a huge number of dendritic spines on it, and they're all going to be producing input. The axon terminals that synapse onto them are going to be releasing neurotransmitter, which will be activating ligand-gated ion channels on the dendritic spine, which will then be opening and allowing in currents into the cell. Okay, now these currents will either be depolarizing or they will be hyperpolarizing. Okay, and for instance in the GABA-A receptors we've seen that there are two potential scenarios. We can either have that depolarizing current in which you move chloride anions out of the cell, okay, or we can have that hyperpolarizing current in which you move chloride anions into the cell. Now, action potentials are not really generated in the dendrites, okay? They are generated first at the axon hillock, okay? The dendrites have far too high threshold potentials for generating action potentials to actually be able to physiologically generate an action potential. The axon hillock has the lowest threshold potential, okay? So what's going to happen is, for instance, if we have our GABA-A receptor here, which is then opening and allowing chloride anions to come into the cell, some of these chloride anions will diffuse over into the axon hillock, and they will cause hyperpolarization of the electrical potential difference across the cell membrane of the axon hillock, therefore making it less likely that the axon hillock will get an electrical potential difference across its cell membrane up to threshold potential for firing an action potential. So it will make it less likely that the axon hillock is going to fire an action potential. On the other hand, if you have one of these bizarre GABA-A receptor junctions here, where when you open the GABA-A receptor, chloride anions actually move out of the cell, then that will mean that chloride anions will diffuse out of this portion of the cell, out of the axon hillock, to go into this dendritic spine, which has lost chloride anions. Okay, so this time we're going to be taking chloride anions out of the axon hillock. We're then going to depolarize the electrical potential difference across the cell membrane, because we're moving negative charge out of the intracellular compartment here. So just to talk you through this, remember, we've got some electrical potential intracellularly and some electrical potential extracellularly. Okay, if we are putting chloride anions in, coming back to our first scenario of hyperpolarization, okay, we are going to be reducing the electrical potential intracellularly. Okay, so this number is going to go down. Okay, that means that if we're thinking about the electrical potential difference from extracellular to intracellular, okay, this number is going to remain roughly the same, but this number has now gone down. So the amount that we are going to have to go down when moving from extracellular to intracellular is going to go up. So remember, it was originally negative 65. If we're moving this down further, it's going to go lower, basically, so it's going to become hyperpolarized, making the axon hillock less likely to fire an action potential. If, on the other hand, it's one of these rare GABA-A synapses where when you open the GABA-A receptors, chloride anions go out of the cell, then you're going to be removing chloride anions from the intracellular compartment. That's going to raise the electrical potential of the intracellular compartment. We're not really going to have that much change to the extracellular electrical potential. So, if we ask now, what is the electrical potential difference from extracellular to intracellular going to do? Well, originally it was negative 65, i.e. we had stepped down by negative 65 millivolts. So in this picture here, here's our electrical potential extracellularly, here's our electrical potential intracellularly. But we've now raised up the electrical potential intracellularly, so the amount we'll have to step down will be reduced, basically. Okay, so that's therefore going to produce a depolarization of the electrical potential difference across that cell membrane, making it more likely that the axon hillock fires an action potential. Okay, so that's how those two different types of GABA-A synapse can work. Okay, and it's not the case that a neuron will always have a GABA-A synapse that will be inhibitory or excitatory. You know, changes in the chloride anion concentration gradient across the cell membrane can change whether the GABA-A receptor synapse is going to be inhibitory or excitatory, but most of them remain inhibitory. Okay, so for the most part, what will happen then is the axon terminal will release GABA onto the dendritic spine. This will open GABA-A receptors, chloride anions will come into the cell, they'll diffuse into the axon hillock, hyperpolarizing the electrical potential difference across the cell membrane at the axon hillock, and therefore making it less likely that the axon hillock will fire an action potential. Okay, so that's why it's an inhibitory postsynaptic current. It inhibits the neuron from firing an action potential. And that's why GABA is called an inhibitory neurotransmitter. Right, I also just want to mention extrasynaptic GABA-A receptors. Okay, so most GABA-A receptors will be on dendritic spines. They'll be located on this postsynaptic membrane of the dendritic spine. Okay, but you also have some GABA-A receptors which are not located on postsynaptic membranes of dendritic spines. They're just located all over the neuron, basically, on the cell body. Okay, these so-called extrasynaptic GABA-A receptors, these respond to ambient GABA that's in the extracellular fluid. Okay, so some GABA will be present within the extracellular fluid that bathes the neurons. Okay, and this concentration can change, okay, depending on different things. And this ambient GABA will be acting on these GABA-A receptors that are located extrasynaptically. And most likely, when they open, because of the GABA, it will be allowing chloride anions into the cell. Okay, and these chloride anions then will also be contributing to inhibiting the axon hillock from firing action potentials. So this allows the ambient GABA concentration to influence how excitable neurons are, and there are investigations into how important this might be in certain mental illnesses. Okay, so potentially too high ambient GABA concentrations could be involved in certain mental illnesses. Okay, right. So we'll finish there for this video. I hope you've learnt a lot about the GABA-A receptors from that.